Welcome to the PharmaSource podcast. Today's episode is the first of 2024 and the start of a new season where we speak to the innovators and decision makers pushing biopharma forwards. I'm Luke Bilton, and in today's episode I speak to Markus Gershater, Chief Science Officer and co-founder at Synthace. We talk about the Synthace digital experiment platform, powerful software that helps to speed up R&D experimentation by helping unpick biological complexity. And we discuss their recent State of the Industry report, which is all about lab automation in life science R&D, a report that really highlights the challenges scientists face in managing experiments.
The fundamental problem we're trying to solve is to how to experiment with something as complex as biology, right? There is no other field that has quite this level of complexity to deal with. Like biology is the most complex system in the known universe, and we have to try and work out how we can then unpick that complexity, unweave it so that we can actually understand what's going on and make much better therapeutics or biological solutions as a result. So that's the fundamental mission, the very high level mission. And we started out actually as a biology company, right? So we have labs in London, that's kind of a testament to our background. And essentially what we were trying to do is to engineer biological systems to produce products. And we were using sophisticated experimentation, so very high dimensional experimentation to do that, which is dramatically more powerful than conventional experiments. But it's also very, very difficult to carry out those experiments. And we started building our own software to help us plan, run, and analyze those high dimensional, very sophisticated experiments. And long story short, that's what's become our main opportunity now, is we turn that into a product, and we're now essentially a software company that is offering that capability to the market.
For someone like me, How would you explain high dimensional?
So the way we were all taught science at school is that you have to hold everything in a system constant, except for the one thing that you're trying to investigate, because otherwise you can't tell which things are having an effect on your system. If you're changing too many things at once, then how do you know which one of them has been responsible for the change that you observe? And this is kind of the introduction we have to like a rigorous scientific method. And then our education in experimental design doesn't go any further than that, right? So we do one factor at a time. The issue with that is that you can't then see if the best level for one factor depends on the level of another. So if my best pH depends on what temperature the system's at, then you'll never see those interactions between factors because you're only ever measuring them one at a time. So essentially what multifactorial experimentation is just a very systematic way of looking at lots of different things simultaneously, but in such a way that you can still deconvolute which ones are having the effects that you're seeing. So it's essentially a rigorous mathematical framework that you can use for doing that. And what we found, because biology is an emergent system, it's inherently massively interconnected. It's often those interconnections which lie at the heart of trying to understand a particular biological system. And you just can't see those interconnections if you're doing things one factor at a time, because you can't see the connection between two different factors or more. So multifactorial essentially just allows you to unpick those interconnections which are fundamental to the nature of biology. So it's a much more powerful way of learning about biological systems and optimizing those biological systems.
And so your platform helps with that process, does it? Helps to unpick it all?
It definitely does. So it helps to run the complexity of experiment that you need to do to unpick all of that. But I mean, it's essentially, so we call it a digital experiment platform. So this is essentially where, instead of the scientist having to think of all of the details, plan out exactly what they're going to have to do in the lab, work out how to do it. Maybe if they want to use automation, they have to program the automation. They have to organize all their samples. And then when they finally get data, they have to wrangle it all together. It's usually copy pasting in Excel, frankly, until they get to a point where they can then analyze it. Well, actually, the digital world can do a huge amount to help essentially just automate that planning, execution, and analysis process. So that's what our digital experiment platform does. And because it's lifting that heavy weight of all of that planning and complexity, it means that the level of sophistication, the level of power of experiment that's now possible is lifted dramatically, because it means that you're relying on digital tools and automation to do a lot of what the scientist was having to do themselves, either in their head or with their handheld pipette.
Where have you seen there being some interesting applications or examples you could share perhaps of where this has really accelerated early stage drug discovery?
We've seen some really exciting results, and frankly it's a testament to what happens when you give a powerful tool to some very innovative and hardworking scientists. So AstraZeneca have actually presented with us at SLAS, so that's the Society for Lab Automation and Screening. They have 2 conferences annually, and they presented with us there about what they were doing. And essentially it means that what they can do is they can do experiments which get them to the optimal conditions for a particular process. I mean, they were focused on assay development. So this is the process that they use for screening through, millions of different compounds to try and find the one that might be a therapeutic. They use an assay for that. And they have to develop that assay first. And that can often take, 9 months to do, or thereabouts. And they found that they could do this dramatically quicker by just basically testing lots of the different parameters simultaneously instead of doing them one at a time. That's essentially the heart of it.
So going down from what would have taken 9 months to how long would you say?
So, I mean, we've seen across our customers that it can be sort of a 30 to 70% time saving on developing a new assay. And that got us really excited, because this is a process that's on the critical path for drug discovery. So the fact that we can compress it dramatically, and there's also great evidence that you actually end up with a higher quality assay at the other end as well, because you have so comprehensively investigated that whole space in these very powerful experiments. So to give you an example, one of the experiments was had 1,536 different unique conditions tested in a single run. And because of the kind of technologies they're using in early discovery, this is in a 1,536 well plate. So each one of those tests is, around 4 microliters. So, you're actually looking at a total volume of an experiment of whatever that is, about 6 mils. Right. So, that's less than the sort of dregs left in your coffee cup kind of thing to completely map out a really complex scientific process. So yeah, it's super exciting to see what our customers are doing with this kind of power that we're giving them. And it feels like an entirely new way of navigating biological processes.
Yeah, in an industry that is so focused on racing to release innovative drugs, time is money. So saving 70% or so is huge in terms of the time.
Yeah, I mean, obviously that's fantastic when we see that level of savings, and it's particularly powerful wherever there's the most complexity. So if you've got a fairly simple assay to develop or media to develop or whatever, then actually there's relatively little you can squeeze out of that, right? But the discovery world is going for ever more complex problems. They're going for ever more difficult targets and ever more complex modalities. And so we're really excited about the way that this kind of experiment platform can try to transform the way that kind of research is being done.
Very interesting. Onto the report then. It seemed to me that there's a healthy dose of realism, I think, coming through on the report. So those 250 scientists that you went to as part of this survey. I really liked the quote actually at the start of the report where you said, even with the best of talent and intentions, it's a complex, messy, and fraught with risk. Why is that? And what are the challenges which are coming through from the report, would you say?
I think it all reflects the complexity of the systems we're trying to work on, and hence the complexity of just the R&D process that's arisen as a result. So if you think about what it takes to actually do any given program of scientific work, then there's a whole ecosystem of different tools. There's a whole myriad of different tasks that you have to do in any given experiment. So inherently, it is super messy. And, the way that we have to do this experimentation is essentially it's all relying on the scientist. It's all relying on the scientist to understand how to interact with all these different bits of software and hardware. It's reliant on the scientist to work out, okay, what might be my best experimental design, and how am I going to lay this out on a plate? How am I going to perform all of these actions in the time that I have available in order to try and make it work? And so they carry huge weight on their shoulders. And then inevitably that means that they're limited in what they're able to carry out. So yeah, there's 87% of the scientists in our survey said that the main challenge with running experiments is keeping track of all of the different elements involved. And so it means, with the best will in the world, these scientists have to be, really intelligent, incredibly organized, really stoic, frankly, very physically coordinated, because they have to, get stuff done in the lab. And then, incredibly analytical with how they then go through all of their different analyses. And so, it's a pretty rare breed who actually manage to then make progress with the biosciences. And then you're still faced with the complexity of biology, which can, take even the very best scientists and, humble them, essentially. 90% of the time, your great idea that you've gone into the lab with is likely to be wrong. And so that's it just adds up to a very messy problem, where even if you're exceptionally good in the lab, it's going to be difficult progress. I mean, frankly, personally, I was never very good in the lab, and that's mostly because I had some but not all of those qualities. I didn't have the attention to detail that is required to really get to that kind of level of meticulous research. I had to force myself into it,
and resilience as well if your theory doesn't work out.
Or that time when you're working in a 384 well plate, and then your colleague comes up and says, did you remember to order in X, Y, or Z? And you're like, no, it's just in the store cupboard on this shelf. And then you go back to your 384 well plate, and you've got to try and remember where you got to with pipetting minute amounts of clear colorless liquid. And the number of times that you just end up sighing and throwing it in the bin and starting over is a little bit too often. And so these are the realities of doing science on a day-to-day basis. And then so when you see that people have difficulty either understanding or completely trusting the data to make decisions. So we had 43% of decision makers saying that they didn't have complete confidence in the experimental data in order to make critical go, no-go decisions. It's not really all that surprising, because it is a hugely tough endeavor that we're undertaking.
Yeah, one of the other stats that jumps out from the report is 90% of decision makers are saying that it's the hardware, the software, the integration between them, which is a barrier. Why do you think that is?
So that was specifically a barrier to digital transformation. So I think we all have this dream, or a lot of us have this dream in the space, that given the complexity of what we're trying to address, that we should be able to get so much help from digital tools, automation, AI, I think, I'm convinced that AI is the future of biology, that and specifically an augmentation of human intelligence with artificial intelligence. So it's working as closely as possible together to navigate biological complexity. But we're only going to get there if we have effective digital transformation of the processes that we're doing. Now, so that's the aspiration. Now, the reason that, hardware software integration is so difficult is simply that there are just so many different aspects to running an experiment, so many different bits of hardware that you might want to do any given experiment. And there's a huge ecosystem of providers which make all sorts of incredibly capable hardware and software. But then that's all really fragmented. And over decades, people have been focused on making ever better instrumentation for doing more and more powerful analyses, for example, but they haven't necessarily been focused on, okay, how can we make sure that the data that comes from these is all harmonized in such a way that can be brought together in a really coherent and automated way. And similarly, on the software side, there's so many different aspects of running an experiment, from planning and resource management, sample management, going through to working out exactly how you're going to do the experiment, the software that then interacts with the hardware, and then doing all of the reporting and analysis. Like each one of these has different software which is best at doing that. There's no kind of one software which combines all of it. And so it then becomes an integration problem there as well. And again, there are no standards. There's nothing there which says, okay, this is the way that the whole industry is going to work together. And there are standards efforts going on, but they are very difficult to make stick, and like they're making slow but steady progress. But in the meantime, we've got this huge integration problem of how we can get all of these things working together in a coherent ecosystem.
Where have you seen teams, customers you work with, or those that you've seen in the industry who are actually getting to grips with this and are making practical improvements to how they manage their data, starting to apply machine learning?
It is becoming more and more prevalent. What we see works very well is when people are focused on a very specific application area where they've worked out that they'll be able to get clear scientific benefits from these kind of things, and then they start in a relatively constrained way and then build out from there. I think what can often be very difficult, and a lot of the industries learn this, is to go for sort of wholesale grand digital transformation projects, where like they have all of the very best intentions, but if they are going to take 3 to 5 years to implement, then often the realities of the dynamics within certainly larger companies means that these initiatives tend to lose the patience and backing well before they would actually produce. And so it's these smaller, much more focused ones that work well in the first instance. And then in terms of specific examples, I think there are some really notable examples in kind of smaller biotechs which have grown up with these kind of philosophies from the outset. So I'm thinking about companies like Recursion and In-Sitro. What they're doing is they're building out these digital and automated systems from the outset with AI as the intent. And so like they built it kind of from the bottom up, so they don't have that same kind of big transformation and change management that might be needed in a more established company. And so they get these fantastically curated rich data sets which then act as the foundation for then artificial intelligence and cutting through that biological complexity in a much more effective way.
A moment ago, Markus, you started talking about a grander vision for what this could mean to humanity. How do you see the role of Synthace within that? I mean, if you could project a few years into the future, how would you like to see the product developing?
Yeah, what we see is a lot of digital tools that are there to help people to analyze their results. And, a lot of AI is being used in exactly this way, or to make better predictions. But what we don't see is digital tools for actually helping to drive experiments. With our digital experiment platform, we're looking to make basically like an experimental engine, if you like. So in the same way as there's a plethora of different tools that you might use for analysis, then there should be experiment platforms allowing you to do experiments in a much more powerful way. And then it's that engine which will then drive the creation of all the very sophisticated and powerful data, which acts as the foundation for all of the artificial intelligence ambitions that we have as a field going forward. So we've got customers now saying they'd like Synthace to be the system upon which all of their experiments are run. And that then means that they can do more powerful experiments, but it also means that all of those experiments are fully digitized. So you have a complete record of absolutely everything that went on in that experiment. So every data point has the full context of exactly how each one of those data points was produced. And that means that you end up with dramatically more useful and robust data sets going down the line, because everything's digitized by default, as opposed to the way that things work at the moment is often that scientists, again, on top of everything else, they're given the burden of recording everything they've done in meticulous detail in some digital repository, which frankly is a bit of a conflict of incentives, because the companies asking them to get on with their science as rapidly as they possibly can. Like you said earlier, it's a massive race to try and get therapeutics into the clinic as fast as possible. But then at the same time, they're saying, oh, but while you're doing that, can you also record everything in a huge detail in these digital repositories so that we might be able to use this data in some undefined future with some AI algorithm that we're yet to think. And so you can imagine what the scientists focus on. They focus on getting things done super quickly, as best as they possibly can, and they won't, spend a lot of time doing all of that digital record keeping. So I think, those kind of digitization initiatives that rely on the scientists to record everything are doomed to fail, because there's always going to be a much more pressing imperative, and that is get that drug through the pipe as quickly as possible.
And once you've got all the data in there, does that open up the opportunity for conversational LLMs or different ways to interact with that data set?
Yeah, definitely. I think actually LLMs offer all sorts of opportunities in terms of that interface between the scientist and technology, or the scientist and particular data sets. And we did a proof of concept where we, in our R&D function, we looked at having an LLM on the front end of Synthace.
Oh, interesting.
And so you can essentially then just ask, tell me a good media for growing T cells is the example we use. And then, obviously, because these LLMs are kind of spookily good, they come back with a very good media for growing T cells. And then you can say, okay, design me an experiment to optimize these different components, the concentration of these different components of the T cell, and it will immediately come back. But it'll also come back with questions of the bits of information that you haven't given it yet. So so fill in all the gaps, and then when it's got the gaps filled in, it can automatically then generate a Synthace workflow, which can then go and be done in the lab. So it means that you have a hugely intuitive interface to what is a very powerful technology. And then similarly, you could see exactly the same thing happening with, interrogating complex data sets and really trying to understand what's going on there. The critical thing is, though, is that, as we know, LLMs, they don't have any particular rigor. Right, they're incredibly, in quotes, creative and, in quotes, knowledgeable, right? But they will also do stuff that's completely illogical. Like, if you see an LLM playing chess, it will take its own pieces, because it doesn't have that logical framework. All it's doing is it's predicting what word should come next each time. That's how these models work. Because Synthace understands what works in the lab and what doesn't. It's got all the models and the heuristics for working out how lab processes work. Without that, then an LLM is likely to do the sort of lab equivalent of pouring a pint into a shot glass. It's not going to come up with logical things that make sense. It has to have that rigor and that check against it. And I think that's what we're going to see emerging, is we're going to sort of see these knowledgeable systems, if you like, these rigorous systems paired with the kind of, as I say, in quotes, creativity and knowledge of LLMs. And that's where it's going to get really powerful.
Fascinating, isn't it? Markus, how about yourself personally? I mean, do you find this work rewarding? I mean, how do you feel about the impact you're making?
I couldn't imagine a more important thing to be working on. I feel immensely privileged to be in the position I am, with the opportunity to try and make these kind of changes in the world. To talk a little bit grandly for a second, I think that biology is the technology which has the most potential for transforming the things that matter to us the most. Whether that's healthcare or food or production of materials and chemicals, it has a huge amount of potential to help us with these massive global problems. But at the same time, it's exceptionally difficult, as we've already gone through in length, to actually understand biological systems well enough and work with those biological systems to effectively solve these problems in any kind of decent timeline. Building these new powerful platforms for enabling biologists to achieve their aspirations that much more effectively is really exciting. And, it's a very, very tough thing to do, to boil down all of this complexity into, a tool that essentially allows you to run these experiments dramatically easier. It's not been an easy path at all, but it's immensely worthwhile. And when I look at what we've achieved, and when I look at the kind of science that scientists are doing with our tool, it never gets old.
Markus, thank you so much for your time. Pleasure speaking to you.
Likewise. Thanks very much for having me on.
I hope you enjoyed the episode. For more insights and best practices, head to pharmasource.global and sign up.